you might notice something looks a little bit different. And that is because I am using new technology in the whale room. I'm using my cheap webcam on the old projector cart, going over chords. Now I have presets, but this camera is way better than my webcam, but it is mounted on a tripod and it doesn't move, okay? So I'm just gonna operate it from the left side. This is where I usually start. And then the center. And then the right side. That's slicker than snot, ain't it? Okay. This lecture, we got to get moving. And the reason why we're kind of moving at a really good clip here is because we lost 2 lectures with 2 Monday holidays. We lost Martin Luther King Day and we're losing next Monday. Okay, so we're kind of got to catch up a little bit. We're still a little bit behind. But what we're doing today is we're finishing lecture exam 1 material, and that's on that board. And we have to start lecture exam 2 material, start it today, do Wednesday lecture entirely on lecture exam 2 material. Then next week, Monday is a holiday, no school. And Wednesday, we have lecture exam 1. Okay, that's a week from Wednesday. But because we don't have school on Monday, we don't have labs next week either. So again, this is a reminder. Please pay attention to these announcements and reminders. Next week, the short week after the President's holiday, you need to return lab kit 1 and pick up the second lab kits. And that's lab 6, 7, and the iWorks Reach DL unit. Okay, that's got to happen next week. I'll do a Canvas announcement with the hours on it. Or if you have the lab 1 piece of paper, it tells you the hours and the locations. Okay, so that's got to happen next week. Or other than that, we don't have lab. You just got that and lecture exam 1 on Wednesday. All right, let's get going on this. Remember, we've finished talking about cell metabolism and how our cells use the 3 nutrient links to make ATP. Now we're on the second big topic, which is DNA. DNA has 2 subtopics, and we're almost done with the first one, and that's protein synthesis. So this is the second big topic, DNA. We've finished pretty much talking about protein synthesis. We covered some preliminary basics like DNA structure, nucleotides, the difference between DNA and RNA, the 3 types of RNA. Then we went through the protein synthesis sequence of transcriptions first and translation. And then I ended last week talking about gene regulation. Everybody on the same page? Now the one last topic we got to talk about is mutations, and then we can go to the last topic of, okay, hold on. This is the pain in the butt here. Just give me a second. Now I'm going to be looking off to the side a little bit because I'm seeing your faces on this huge, big screen TV. Okay. Here we go. This is a lot of this information so you guys don't get lost, you know. So I'm giving you this housekeeping information verbally. It's on this first slide. We are finishing lecture exam 1 material. We're starting lecture exam 2 material. Next week, there are no labs because of President's Day on Monday. Everything I said, okay? You got no excuses. All right, so last topic is on mutation. Mutation literally means a change from the norm in nucleotide sequence. So remember that all our proteins are coded by genes in our DNA, and that's DNA nucleotide sequences. Well, if there's a change in that, that's called a mutation. Now this is a huge topic, and here are some highlighted words. We're going to mention one germline mutation, but we're basically talking about somatic germline mutations. And I'm going to write this over here on this board. All right. There are, see, isn't that weird? Even though the camera's way over there, you guys can still read this, right? Yeah, so that's why it works. Okay, we got germline versus somatic. Somatic are basic body cells. Germline cells is in your gonads, the cells that make spermies and eggies. Those are germline cells. We're going to be talking about basically somatic mutations. And then there are chromosomal mutations versus gene, or what is sometimes, I need a new blue pen here, sometimes referred to as point mutations. Can you guys see that in your screens? And I underlined basically what we're going to be talking about. So I already explained the difference between germline and somatic. Gene or point mutations are 1 to 3 nucleotides change. Chromosomal, you can actually see it, and we'll talk about this in just a little bit when we talk about DNA replication. We could see it in a karyotype. And if you take a picture of a chromosome and you could see it change that big in a microscope because the chromosome has changed that much. We're talking about hundreds of thousands to millions of nucleotides. Can anybody give me an example of a chromosomal mutation you can actually see in a karyotype? Some of you have had some biology. Anybody? You can actually take a picture of a person's chromosomes and it's messed up. You can actually see it's messed up.
Would Down syndrome be?
Down syndrome. You could actually take a look at the chromosomes. You got 3 chromosome 21s, trisomy 21. That is Down syndrome, okay? You can actually see it. That would be a chromosomal mutation. But we're gonna spend our time here mostly talking about this. I will mention one germline gene mutation, all right? And so that's explaining this first bullet. I even put those other words on here. Okay. Now, most of the changes that happen in the chromosomes, let me go back over here. Oops, something got messed up here. Okay, you'll never notice. And there are 3 billion base pairs, and I said every time that we replicate our DNA, on average, 1 gets erroneously read. And that is extremely accurate when you're talking about 1 out of 3 billion. But when that happens, you don't notice it. And the reason why you don't notice it is because of the second bullet, right here. Most of our, remember, less than 2% of our DNA codes for anything or regulates anything. It's just space filler. That's over 98% of space filler. If you got one nucleotide that changes in a space filler, you never notice. Another reason is this third bullet. Now, you do not have to memorize the tables of codons for lecture exam 1. That's not the point of this class. But if you look at these, and your textbook has one, tables tend to look like this. Now, remember that from last lecture, codons are on our messenger RNA. So this is RNA, that's why you see U's and not T's. You guys remember that? Okay. Well, if you look at this on this side, the RNA nucleotide is the first of the triplet. Remember, they come in triplets. Across the top is the second. And over here on the right is the third nucleotide of the triplet. Now, there are 20 amino acids that go into all our thousands and thousands of proteins. We only use 20 different links, okay? Now look at this carefully, and this is the fourth bullet. Things like leucine and proline and arginine, they're actually only coded by the first two nucleotides. If the first two nucleotides is CU, it don't matter what the third nucleotide is, it's going to code for leucine. If the first two nucleotides is CC, it don't matter what the third nucleotide is, it's going to be proline. Now, not all of them are like that, but 8 of them are like that. Can you see that in the tables? And if it's not a full codon redundancy, some of them are 2. If it's UA, you've got a good chance of being tyrosine, and it could be the third nucleotide could either be U or C, don't matter. Everybody see that? So there's some level of redundancy pretty over all the table of codons. If you get a point mutation and you change the third nucleotide, you won't notice it for those amino acids because it won't change the amino acid. You got the same protein. Everybody see what I'm saying with codon redundancy? So if you get a point mutation, one nucleotide changes, and it's in space filler, nothing, nada. And even if you do get a point mutation in a gene, if it's the third nucleotide, there is a decent chance, about a 1 in 4 chance, it will be also nada because the amino acid is unchanged. Questions on that? But it's got to be the third nucleotide. That's why it's embolded. If it's the first or second nucleotide, it's going to change the amino acid and you're going to get a screwed up protein. Okay? And because of specificity, you can just change one amino acid out of hundreds and you can screw up the protein because of specificity. Okay, and that's based on 3-dimensional shapes. And this is why I'm giving you one example of a germline point mutation, and that is sickle cell anemia. Okay, that beta chain has 286 amino acids, and this is what I was talking about last lecture. I said, let's round it up to 300. How many nucleotides? 900, because it's 3 to 1. You guys remember that? Okay, well, in this case, look at this guy here. We're changing the second nucleotide, and that for sure changes the amino acid from glutamic acid to valine. Now, you don't need to know that specifics, just know that sickle cell anemia is an example of a germline mutation that actually does change the second nucleotide in the gene. That changes the amino acid. Now, in the beta chains, we'll look at this later when we talk about blood, valines tend to stick outside the edge of that glob, like this, like my arms. It sticks out. And valines tend to stick together. And when the oxygen tension gets low, oxygen levels drop, they start sticking together. And here's one quaternary hemoglobin, and you better know what that means for lecture exam 1. With the betas chains, with the valines out like this. Well, the next one is going to stick to it. And the next one is going to stick to it. And each one is sticking, and it forms this huge polymer of hemoglobin. And because of the shapes of the beta chains, it can't form a straight line. It forms a real gradual curve. And when all the quaternary hemoglobins in a red blood cell is forming a real gradual curve, the red blood cell changes shape into that. That's why it's called sickle cell anemia. Any questions? And then these spiky sickles get stuck in capillaries, and it's extremely painful, and tissues die, and eventually you die. Any questions on mutations? All right, so that's it for protein synthesis. Now we're going to go to DNA replication. Okay, so we're coming to the last topic of lecture exam 1. We're finished with this. Now we're going to DNA replication. First, let's talk about differences between DNA replication and protein synthesis. Well, one of the things is we got differing enzymes. But just remember that DNA is a double helix, and your regular somatic cells, your regular body cells, only have 46. So you're going to have enzymes, and if you have your lecture packet, I forgot to bring it because I got so much on my mind with this new system. I think it's page 16. It talks about some of these enzymes and how it contrasts with protein synthesis. You're going to get a helicase, and this is down there in the bottom of page 16, that actually unzips the double helix. It's also got to do that in protein synthesis too, but in the case of DNA replication, eventually the whole double helix has got to unzip. Here, it's only got to unzip at the gene. Okay. But if you look at this animation and look at last week's animation on protein synthesis, it looks almost exactly the same, doesn't it? The all nucleotides come in here as triphosphates. However, in this case, instead of making messenger RNA, this is a DNA complementary strand. But it's also doing the same sequence. Hydrogen bonds first, then the covalent bond we call the phosphodiester bond that you have to visually recognize on lecture exam 1. The reason why it is DNA nucleotides is because the enzyme is DNA polymerase. For protein synthesis, it was RNA polymerase. This is DNA polymerase, and it's going to hook up complementary base pairs of DNA nucleotides. Okay? But each one is a triphosphate. So if you got 3 billion base pairs to reproduce and replicate your DNA, it's going to take 3 billion ATPs. If you look at this animation, you're going to understand why. Okay. Any questions? All right, now this, what I want you to write on the slide on the bottom right for this animation that looks like protein synthesis, I want you to write leading strand. Now, those of you who took me for anatomy, we covered this in anatomy. We're going to talk about semi-conservative replication. but I made it really simple in anatomy because most of you are like going, oh, what is this? I don't even know which way's up. So that's why I did it really simple DNA replication in anatomy. Well, I got to introduce another level of complexity. And believe you me, this is actually more complicated than this. And that is this. What you're going to write next to that animation on the previous slide is leading strand. The leading strand looks like transcription to make a messenger RNA for protein synthesis, and only the leading strand. Now this is this figure from your textbook is showing you really good. Now remember I told you that DNA is not a mirror image. It's not a left strand meeting a right strand. You guys remember that? It is all left, which means this strand, oh, this is going to be hard, is going to meet this strand upside down. Well, here's the thing with DNA polymerase. It can only read from the original strand from the head to the toe. You guys remember me telling you this? If it's all lefts, here's my head, down there on the floor is the toe. If the other one is upside down, my head's on the floor and my toe's up on the ceiling. And DNA polymerase can only read from head to toe, or from the 3-carbon to the 5-carbon. So the original strand, here's the head, 3-carbon, and down here's the foot, the 5-carbon, and DNA polymerase can only read in that direction. That's the leading strand. Now this is all discussed on page 16. For the other strand, because it's a double helix, again, here's that unzipping enzyme helicase. It's going to unzip the double helix completely. It has got to do it this way. It has got to go from, because here's the foot of the original strand, it's got to go from the head to the foot, jump, head to the foot, jump, head to the foot, jump. And now you're going to get DNA fragments initially. DNA fragments initially. And that is called Okazaki fragments, because this is the Japanese scientist's name who discovered it. Then there's going to be a third enzyme called ligase that is going to tie these Okazaki fragments together. But the reason why there are two different patterns of replication is because the enzyme can only read from head to toe. And one strand is right side up, and the other strand is upside down. That's the simplest way that I can explain that. Any questions? It's all on page 16, and this figure will give you a really good indication of how it's doing it. It can only, so we're talking about the original strand, head, 3-carbon, the toe, 5-carbon. Here's the toe here for the other original strand. It's got to start here, go to the toe, jump, go to the toe, jump, go to the toe, and that gets you Okazaki fragments. Then you need a third enzyme only on the lagging strand to tie them all together, and that's ligase. Any questions? This big TV is outstanding. You can actually see your face. So when you're on your webcams, don't think that I can't see that you look really bored, because now I can really see it with this huge screen TV. All right, any questions on this difference between leading strand and lagging strand? And on this animation, this is going from head to toe. This is the leading strand. Any questions? Okay, well, this means if you look at that previous slide, The faint colored strands are the original strand, and the complementary new strand is in dark purple. Can you see that? The faint, more like pink purple, is the original, and the new complementary strand is dark purple. That means if this is the original double helix, now we call these two the daughter double helices from DNA replication. Each is half pale, half dark purple. Half pale, half dark purple on this figure. Each daughter double helices, the products of DNA replication, are half gray, the original, half new, the purple. Everybody see that? And since replication results in half old, half new, that's why we call it semi-conservative replication. In the new two daughter double helices, the old original double helix was conserved, but only the half of the daughter double helices is conserved. Semi-conservative replication. Someone tell me the error on the bottom left of the slide. What's wrong about it? It's great for the way I teach this in anatomy. Insufficient for you guys, but I use it, so I ask you what's wrong with it. The longer you take, the less time I'll have to review for lecture exam 1, which I'm doing today. And if we run out of time, I won't review for lecture exam 1.
Are they both doing head-to-toe replication?
They are. This animation is showing you 2 leading strands, and that is wrong. Only one of the strands should be adding nucleotides one at a time like this. The other one is supposed to be jumping, forming sections or Okazaki fragments. Everybody see that now? Ain't science a bitch? Actually, no, it's bitch in. Because it explains a lot of things, and a lot of things that will help you to function better, even as students. Okay. Let's go on to cell division now. And here's where more of a definition of the difference between somatic and germline. We had this covered in anatomy. You should have had it covered in anatomy. So I should be able to go through this pretty quickly. But regular body cells, somatic cells, only do mitosis. Germline cells do both. It's not that germline cells only do meiosis, it's just germline cells do both. Once they do or get started in meiosis, they're not going to do any more mitosis. Okay? Kinesis in this context means division of. Karyo means the nucleus. And what this second bullet is saying, there is division of the nucleus first, then division of the cytoplasm. What you might want to see that, what you might want to write next to the second bullet is you will see that in telophase. Second bullet, all in telophase. You'll see the nucleus divide first, then after that, division of the cytoplasm, cytokinesis. Cell cycle. And when I teach this, I got this from Questa and I thought it was clever, so I keep it. And that is cell cycle's IP-MAT, right? But if you look at this figure on this slide and look at what I have in green, maybe I should have made it in brown when I change it. Here, I'll do it in colors that are consistent with this figure. PMAT is mitosis. But the whole cycle is IPMAT. But if I were to say it more chronologically correct, it would be IPMAT. The vast majority of a cell's life is spent in interphase. In fact, that's why we split interphase up into 3 subphases. We'll talk about next. Okay, now some textbooks will make cytokinesis its own phase, but I don't because then IP-MAT don't work. So I keep it simple, IP-MAT. And again, it is the vast majority of the slice of the pie. Like that last figure that was purple and brown, they made mitosis way bigger just because they wanted to fit in the letters. It is actually a much smaller slice of the pie, like this figure. Everybody got it? All right, so the subsections of interphase is G1. This is right after the last cell division, the last mitosis. And you're getting a lot of synthesis because the cell's got to grow. To grow, it's got to absorb a lot of fluid, that is pinocytosis. And already the centrioles replicate right after the cell divides. Right after the cell divides, the centrioles that went to opposite sides of the pole, they're in pairs in what's called a centrosome, but boom, they get divided. And so there's only one centriole pair after mitosis, after cytokinesis. Well, right away they replicate. S is where the semi-conservative thing happens. Okay. And then G2 is second gap phase. This is where the cells are getting pretty big. Other metabolic activities start to slow down because they're already big. And the cell is just preparing to go into the next cell division. Now this is different cells divide at different rates. The rate of IP-MAT is very different from cell to cell. Any questions on interface? All right, now to discuss cell division. This should have been covered in anatomy, so I should be able to go through this pretty quick. You need to understand the various Chrome words. Oh, I can finally shift. When the DNA is in interphase, we call it chromatin because we can't really visually recognize the 46 chromosomes, and the 46 double helices, they're all just intertwined. When you get into cell division and prophase, they start to condense when you can actually see chromosomes, separated chromosomes because of their ultra condensed. Now we'll call them chromosomes. And the chromosome will look like an X, like what I have on the whiteboard here, because it has already gone through S in interphase. it has already gone through the semi-conservative replication in S and interphase. So it's replicated. And both copies are attached. And each copy is called a chromatid. So this blue arrow is one chromatid. This blue arrow is another chromatid, or what's called a sister chromatid if it's the copy. And each chromatid is attached at the centromere. Okay? And on the whiteboard, and you can see it in the little picture, it's the black circle. That's the centromere. That's where both of those chromatids are attached. All right, now in mitosis, as it says here on this bottom arrow, this is a side of spindle apparatus, and I'm going to put it in the whiteboard in purple. Here's one spindle apparatus, here's the other. The spindle apparatus is microtubules anchored by the centriole pairs, anchored by the centrosome. And they're at the opposite poles, okay? Everybody got this terminology here? It's in this figure, it's on the whiteboard, and it shouldn't be the first time you're hearing this. All right, let's go through mitosis. In mitosis, all 46 chromosomes are doing this. In prophase, you got the DNA condensing, so now we can recognize them as visual chromosomes. The nuclear envelope dissipates. Then the spindle apparatus gets organized with microtubules attaching from each centriole pair as they migrate to opposite poles to each of the 46 centromeres. In fact, why don't you right here on this bottom arrow right here, you should write 1 centromere equals 1 chromosome. 1 centromere equals 1 chromosome. So it's going to set up something like this. Prophase means like previous, so for the beginning stage of mitosis is prophase. Meta means middle. Now, since the spindle apparatus is pulling equally on all 46 centromeres toward the poles, this pulls each of the chromosomes, each of the 46, onto the equator, the middle of this dividing cell. Also, by the time it's in metaphase, these chromosomes are as condensed as they're going to be, which is why we take a picture of them in metaphase when we're going to do a karyotype for which we can see chromosomal mutations. But we take a picture at metaphase because now they're maximally condensed and they are as consistent in appearance as can be as they're maximally condensed. While in anaphase, the prefix animase, ana means split. And in anaphase, the centromere split because the spindle apparatus keeps pulling really hard. Now, what we used to call chromatids now are chromosomes because each has their own centromere. This is what happens in anaphase. Now look up on the whiteboard if you could see that little picture. How many chromosomes do you see here? How many black circles am I pointing at?
1.
1. There's 1 centromere I'm pointing at. That's 1 chromosome. How about over here?
2.
2. Now do that 46 times. In anaphase, the chromosome count doubles. And if you're going to start out with one cell with 46 to two cells with 46 chromosomes, at some point it's got to double, and it's an anaphase. Okay, centromere split. Now what we used to call chromatids are now newly designated chromosomes, and they're pulled to each pole, and eventually it's going to divide right along this plane. And I'm writing with the dashed line. So if I'm doing this 46 times, here's 46 chromosomes. This one has 46, and this one has 46. The two daughter cells were just like the original cell, 46 chromosomes. At some point, it's got to double. That doubling is an anaphase. Telophase is just the opposite of prophase. The chromosomes decondense. The nuclear envelope reforms around them. Hey, right there in that box for telophase, first bullet is karyokinesis. Look at that box here for telophase. The nuclear envelope reforms around both nuclei. This first bullet is karyokinesis. That happens first. Cytokinesis happens second. And both of those are in telophase. And that's what I said way back here, so many slides ago, right there. This bullet refers to telophase, and it is saying karyokinesis happens first, the division of cytoplasm, cytokinesis, happens after karyokinesis. Karyokinesis first, and that's this first bullet. So write down right here, karyokinesis. Cytokinesis happens after. Any questions? If you're starting with one cell with 46 and you're going to end with two cells of 46, you got to double somewhere, and you double in anaphase. Okay. Here in your textbook, this is only in the older 4th edition. They got rid of this figure. I can never figure out why. But here now that we are now designating them chromosomes, no longer chromatids. Yeah, we go into anaphase with chromatids. We come out anaphase with chromosomes. Oh, I guess I can play this. Let me know how the audio goes with this. Like maybe not playing at all. Hold on. No, it should be playing. Well, just read the words then. There's no volume.
Can you switch it back to the screen, please?
Oh, that's right. Once I check the volume of my thing, I got out of it. Hold on. See it now? All right, great, no volume. Did you guys get the idea though?
So a quick question, Professor, about that. So
Right.
pretty much when we're doing mitosis, it's pretty much like when there's damage to your body, it's beginning to regenerate cells, like heal. But as for meiosis, is for like reproduce or reproduction for the
Yep.
cell?
Yeah, we'll get to meiosis in just a sec.
Oh, okay.
But mitosis is both for repair of damaged tissue and regular cell replacement, even if there's no damage. Cells don't live forever. And so like say in your skin, that stratum basale, you guys remember that from anatomy? Constantly going through mitosis, you slough off the surface stratum corneum, that becomes your house dust. And in the meantime, you're constantly replacing your skin. And that doesn't necessarily have to do with damage and repair to damage. But if you do, like in that video, have damage, you're gonna have mitosis and an increased rate of IPMAT, okay? All right, again, meiosis, germline cells will do mitosis first and then do meiosis. And the meiosis that I'm about to describe is male meiosis. I'm doing male meiosis first because males are better. No, because males are simpler. And we'll talk about female meiosis at the end of the semester. And instead of IPMAT, meiosis can be described as PMAT, IPMAT. And what does that tell you?
Everything but interphase happens twice.
So there's two rows of cell division. PMAT is cell division. So if it's PMAT, IPMAT, as it says right here, there's going to be two series of divisions. You do have to know a little bit about biology, and our regular somatic cells are diploid. That means it has two of each type of chromosomes. N is the number of chromosomes in a species. And N for Homo sapiens is 23. We have 23 different types of chromosomes. But our regular body cells have a pair of each, 2N or diploid. And that's what I wrote down here on the whiteboard for mitosis, 2N. All 46 are doing it. You have 23 chromosomes that come from mommy. You got 23 chromosomes that come from daddy. And meiosis is about making sperm and oocytes. And it involves two rounds of cell divisions. Especially for the second bullet, I might write next to the second bullet, males only. This is male meiosis. Okay, and here's the karyotype, and look at it. This is a picture of metaphase chromosomes. Why? They are maximally condensed. Okay, and they're arranged from largest to smallest, chromosome number 1 being the biggest. The reason why trisomy 21, a lot of those babies survive to term and actually live several decades is because chromosome 21 is pretty small and doesn't have a huge amount of really crucial genes on them. But since you have a number 1 from mom and a number 1 from dad, when you put them together, we call those homologous pairs, or homologs for short. The first 2 homologous pairs are called autosomes. That'll become big later in the semester. And the 23rd pair are the sex chromosomes. Look at this karyotype and tell me what is the gender of the person of this karyotype.
Male.
Male. Now, I'll just mention this now since it's lecture exam 1, but most of the physiology I'm teaching you this semester has been oversimplified to the point of actually being inaccurate at best, a lie at worst. And what students don't understand is this: you take this class at this rudimentary level, we are teaching you lies. Why are we teaching you lies? So that you can understand it, because you can understand it at this level. This right here is making it really simple, right? XX female, XY male. It is way more complicated than that, having to do with estrogen and testosterone receptors in the germline cords. And there's so much more to it than that. What you need to know is that there is no straight male and female. There are 2 bell-shaped curves, one on a male pole and one on a female pole. Everybody got that? Things are way more complicated than people realize, especially when they post on social media. Okay, here we go. We're going to talk about meiosis 1 first, because it is the freak. It is the reduction division. This is where we're going from diploid to haploid, 2N to N. That's what it says right there on the slide. And the reason why it is a freak is because this is the only kind of division in meiosis 1 where the homologs get really close. In mitosis, they're totally random. They don't care where the other homolog is. But here, they get really close. They get so close, they exchange pieces. They're underlined and highlighted in what is called crossing over. You can see it in this animation right here, and they exchange pieces. I only did it once down here. It looks like a smiley face. But here's what, these are both, say, chromosome number 1s, and they exchange some pieces. Everybody see that? That's cross. Doesn't it look like a smiley face? Oh, I'm so happy you're teaching me lies all semester. Okay, anyway. Because of that, the homolog's coming really close. Okay. The spindle apparatus attaches to centromeres differently, because these will actually get so close in the way. From this centriole pair, it'll send the spindle apparatus to just one connection, to the red one, and another different connection to the blue one. That is completely different. So each centromere only has one connection. Up here in mitosis, each centromere has 2. This one only has 1. One's going to the left pole, the other one's going to the right pole. Metaphase 1, they will maximally condense. They will be in the equator, but now the weird thing is in telophase 1. Because of the way the spindle apparatus attaches, in anaphase 1, the centromeres do not split. The chromosome count does not double. Here, just look down here at the bottom. I think you can see it in the tiny thumbnail. How many chromosomes do I got here?
2,
2. How many chromosomes do I got here?
2.
2. They did not double in anaphase because the centromeres did not split. So in anaphase 1, the original cell had 2. Now this cell's only going to have 1, and this cell's going to only have 1. The number of centromeres and the number of chromosomes in meiosis 1 are going to be cut in half. Another way to look at it in this picture on the whiteboard, you can see it in this animation too, but it's really fast, is that in mitosis, the X become 2 lines. In meiosis 1, X's stay X's. Can you see that on the whiteboard? X's stay X's in meiosis 1. It is the freak. Now in this animation, I wish this animation would go a lot slower. But when that one big circle turns to 2, that's meiosis 1. Notice that X's stay X's. Then when it goes from 2 to 4, that's the next slide. I wish this animation went slower, but look at this. From 1 to 2, 4 X's. From 1 to 2, X's stay X's, but it goes from 4 to 2. The reduction division. Telophase is also weird in that there's no reason to decondense the chromosomes, no reason to form nuclear envelopes. It's just going to go into another division. Okay, but there is cytokinesis. The I between the PMAT, IPMAT is interkinesis, and that's just the resting phase before it goes into meiosis 2. Again, this is male meiosis. Any questions on meiosis 1? This is the freak. Meiosis 2 is very similar to mitosis. X's is going to turn into lines. The spindle apparatus is very much the same. It's just doing it with half the chromosomes. So if you look at this animation going from 2 to 4 circles, X's become lines. And each one only has 2 chromosomes. We're starting out here with this, with 4. 4 X's. Meiosis 1, it's down to 2 X's. Meiosis 2, then now each has 2 lines. Again, this is male meiosis. Well, on there at the very bottom, I make a reference to this. This should have been covered in anatomy, so you should have heard it before. But in female meiosis, all the cytoplasm goes to one side and you get polar bodies. Any questions on lecture exam 1 material? Because that's it. I got 20 minutes, dang. Okay. No questions? Again, it'll be, lecture exam 1 will be a week from Wednesday, because that is the only thing we'll have next week besides you doing the lab kit exchange. But because we're running out of days, I have to start lecture exam 2 material now. Lecture exam 1, are you paying attention? We start out with basic concepts. Again, the vast majority of points are going to be metabolism and DNA, but we start with preliminary concepts like homeostasis, negative feedback loops, positive feedback loops, specificity, lock and key, all about 3-dimensional shapes. We got phospholipid, Chemistry, you need to know that. You need to know the fluid mosaic model of cell membranes, the top 5 components of cell membranes, and which ones do we need for selective permeability. You need to know all the passive and active transport processes we went over. What's moving, which ones have concentration gradients, and which way are they moving to the concentration gradients. Are they going through the lipid bilayer, or are they going through protein channels? If a protein channel is involved, it is called mediated transport. And a pump is considered a protein channel. So primary active transport is mediated transport. It is all on the slides, people. Highlighted, underlined, or bolded. You need to know preliminaries for metabolism, like anabolism, catabolism, decarboxylation reactions, dehydration or condensation reactions, hydrolysis reactions. You should be able to identify them. There is some chemistry you have to identify. You should be able to identify those reactions. You should be able to identify ATP and where the high energy bonds are and how much energy is in there. You need to know the 4 biologically important macromolecules. Even though I went over a big section on the 3 nutrients, when I get to DNA, you got to know DNA structure. So like you have to know peptide bonds, visually recognize peptide bonds. You have to visually recognize phosphodiester bonds. You need to know some of the other preliminaries of metabolism about the redox reactions and electron carriers. And you know the difference between substrate level and oxidative phosphorylation. Remember that there's a handout of potential essays on lecture exam 1. It could be, if it's a short essay, just a short essay, and I'm not going to be correcting your English on Canvas. I'm just not going to do that. Just give me the content, okay? It could be a short essay, or it could be short answer, or it could be fill in the blanks, but any of those things that's not multiple choice or matching or true false, it's going to come from those essays, and all the words are there, all the concepts are there. You need to be able to visually recognize glycolysis, Krebs cycle, and the ETS in a figure that's very similar to what I had on the whiteboard. You got to be able to recognize the difference between purine and pyrimidines. Now, there's a lot of things you don't have to know about intermediates. You don't have to know about the intermediates of glycolysis, but you got to know what goes in and what comes out. Krebs cycle, what goes in, what comes out. You need to know that linking or intermediate step as well. In the ETS, you need to know the chemiosmotic theory. It's one of the essays. ATP synthase is another. You also have to know about the final electron acceptor. You need to know which bases are found in DNA, which ones are found in RNA, which ones are purines, which are pyrimidines, and you also need to do complementary base pairing. What goes to what? You need to know which nucleic acids are involved in transcriptions, which ones are involved in translation. You need to know initiator and terminator codons. You need to know the various terms that I went over today on cell division, DNA replication, cell cycle, mitosis, and meiosis. Boom, it's straightforward. I've been telling you so far what you need to know for lecture exam 1. Lecture exam 1 is a test of if you know it or not.
All this is on the study exam, right?
Say that again?
Everything's on the study exam you give?
Study exam. You mean the study guide?
I'm sorry, yeah, that's what I meant.
Study guide is a list of topics. It is not telling you to what degree you need to know those topics. The PowerPoint, if you follow all of this first 4 weeks of PowerPoints, if you follow it, it's following the study guide. So the study guide is showing you a list of topics you need to know. The PowerPoint is showing you the level of detail that you need to know those topics. It is that straightforward.
Got
Any other questions? Nothing tricky. All right, I got 13 minutes. I'm going to use them all. Okay, I bet you probably didn't print up study guide number 2, huh? Okay, well I'm starting it right now. So if you have it on your device, you may as well pull it up. But lecture exam 2 is basically on signaling organ systems. So we're talking about the nervous system. And we have to look at this very differently than we looked at it from anatomy, because this is about physiology. Although if you took me for anatomy, the section when we get to the functional regions of the brain, you guys are way ahead. Because that splitting it up into the 5-lobed brain, same thing here. And I'm probably only adding about 20% of what I covered in anatomy. So if you have that from anatomy, you are way ahead. Peripheral nervous system and autonomic, sympathetic, parasympathetic. We've got to cover that here. And then we're going to go into the senses. We're going to spend really the main time talking about visual physiology and inner ear physiology. And I think even though it says endocrine system, I have your endocrine system in lecture exam 3. Okay? So if you don't have that, that's basically what we're doing. And that's why we're going, you see the slide that's up on the screen right now. And it relates to the picture that's behind me on the whiteboard. I do have to get as far as I can in this. We should be able to go pretty quickly in this. I'm starting out with just a review of anatomy that you should have gotten from anatomy. There are various types of neurons, and we talked about those in anatomy: unipolar, bipolar, and multipolar neuron. There's also anaxonic. What you want to write next to this, the very first slide of lecture exam 2 material, this one right here, is that we are concentrating on multipolar neurons. Multipolar neurons. And multipolar neurons are written up here on the whiteboard. It's also in this figure. Each of those dendrites is a pole. So on a multipolar, we have a whole bunch of dendrites. And it says their input zone. I want you to change something for me in just a second. We also have the cell body. The input zone is both of these bullets. A neuron can get input on both the dendrites and the cell body. So make sure you write that down next to these 2 bullets. One thing that is written on the second bullet, as I remind students from anatomy, because of Nissl bodies, or the chromatophilic substance, which are bluish gray in color, it makes the cell body bluish gray because of the Nissl bodies. If you get a collection of cell bodies, either in the CNS or the PNS, it looks gray. So we call that gray matter. Gray matter is where the cell bodies are found. White matter generally is made up mostly of myelinated axons. Now, it can be myelinated or not, but myelinated axons are whiter. Whiter. And so we call that collection of myelinated axons white matter. So this is gray matter, this is white matter when it's myelinated. Now, a couple things that are going to be really key about a multipolar neuron. And I have it on the whiteboard here. It's not on this slide, but we're going to talk about it more and more. And that is the base of the axon, where it comes out of the cell body, is called the axon hillock. That is a real key spot for multipolar neurophysiology. So a multipolar neuron can have many dendrites, but it'll only have one axon hillock. The axon come out of it, and as soon as it comes out of the cell body, it can divide and branch. It's called telodendria, but I'm just going to draw one axon. And down here is the axon terminals, where we will see a synapse. So here in blue are the key spots for what we're going to discuss in physiology for a multipolar neuron. Okay. Does this ring a bell from anatomy at all? God, I hope so. Okay. And this is very similar to what I have on the whiteboard, because I went ahead and drew two multipolar neurons, showing you that where one ends and the other begins, there's a small gap. They don't actually physically touch, but they get really close together, and that's called the synapse. That should have also been discussed in anatomy. Okay. And it's showing you here the axon hillock, like I have on the whiteboard, and the axon terminals. Okay? And yes, the signal is going in this direction, out of the cell body. Again, from anatomy, remember that the CNS is the brain and spinal cord, and the PNS, peripheral nervous system, is just everything else. I told my students that this is a gross anatomical distinction. It is not a neurological one. The same neuron can be in both. But if I have a cadaver out with the nervous system dissected, I can put a pin on the CNS, and I can put a pin on the sciatic nerve. That would be PNS. Okay. Also, you'll see these terms afferent and efferent. Afferent means sensory, efferent means motor. And that should give you a help on English papers between whether you should talk about something being affected or something being effected. It's the same root words. I will talk about this later, not today, but these terms, afferent and efferent, are PNS terms. In the CNS, we use ascending for sensory and descending for motor. Those are CNS terms. Don't worry, if you don't get it now, I will cover that again later. Now let's talk about some of the neural glial cells that help neurons function better. These is going to be very key. And since you've had it in anatomy, I'm going to go over this pretty quickly. The myelinating neural glial cell in the PNS is the neuroleumocyte or Schwann cell. It is dedicated to a single axon, just about a millimeter of it. It is like the faithful wife. Here is the oligodendrocyte, the myelinating cell in the CNS, because there are so many axons packly densed in the brain and spinal cord. An oligodendrocyte will branch and myelinate several axons. Faithful spouse, the whore. Now this is what you got to understand too about, I know it's so funny, you know, I mean, I watch this screen on all your faces on the big screen. I can tell who's listening or not. A lot of times I just say nasty shit just to see if anybody's listening. I can tell when you're not. All right, now when you look at this, what you need to know is that all of the axons are in neuroleumocytes in the PNS, all of them. if they just sit in the neural lemmacyte, this is an unmyelinated axon. It's still right next to it. It's just kind of sits in it. But when a neural lemmacyte wraps itself over and over and over again, this is what is myelinating the axon, because now it's not hardly any cytoplasm. It's almost all membranes, phospholipid bilayer, which when it gets really condensed like that becomes really waxy. That's what we call the myelin sheet. Okay? And both the Schwann cell neural lemmacyte, which is only on one axon, or the oligodendrocyte are forming these structures. The other ones in the CNS, so the only one that's not in the CNS is that neural lemmacyte. That's PNS only. Oligodendrocyte is CNS. That's the first one. Here's the second one, astrocyte. Forms a blood-brain barrier. It's the most numerous of the neuroglial cells in the CNS. And since it contributes to the blood-brain barrier, if you see it wrapped around blood vessels, you know it's an astrocyte. Again, we already mentioned oligodendrocytes, the myelinating cells in the CNS. Because of the blood-brain barrier, white blood cells cannot get out of the blood vessels into the CNS. So you need to have phagocytes in the CNS to make up the gap there. Whereas white blood cells can go everywhere in your body, it can't go into the CNS because of the blood-brain barrier. So you have phagocytes there that are fixed in the CNS called microglia. And then the last one are ependymal cells, and ependymal cells are all about cerebral spinal fluid, CSF. Does this ring a bell from anatomy? Yeah. Ependymal cells make it, they monitor it, they maintain it, and with little cilia, they circulate it. And that's the picture of one right there, circulating the CSF. And there's a picture there. Again, if it's lining CSF, it's got to be ependymal cells. If it's lining a blood vessel, it's got to be an astrocyte. If it's myelinating, it's got to be an oligodendrocyte. And if it's none of those, it's got to be microglia. Okay. I'm not going to get to this time, but I'm going to bring this up starting on Wednesday's lecture, this term membrane potential that's on the slide. Remember, for lecture exam one of the short essays is the different ions in the ECF and ICF. Does that ring a bell? ECF and ICF. Not only are there different ions, there are different concentrations of them. Since some of them are positively charged, some of them are negatively charged, this causes what's called a membrane potential. And you think, well, membrane, what does that mean? Well, it's right here on the whiteboard, so get it. Membrane potential doesn't mean, oh yeah, this membrane could be a nurse someday. No, that's not what it means. It means voltage difference across a plasma membrane. You got to get past thinking of potential like how most lay people, patients, think of potential, and start thinking of membrane potential like a healthcare provider. It's the voltage difference across a membrane, and it is because of those different ions and different concentrations. Okay? Any questions? I'm going to end now. Just as a reminder, way on the far right, I got some times. This is when you want to log into the Zoom meeting. You want to be on Canvas before then. Get all that stuff done that was in that announcement that I went over last week. But then log into the Zoom meeting about this time, LAP and everyone else. But this is your actual lab quiz 1 times. It will show up on Canvas at these times, but you have to turn it in. And you have to turn it in at the same time because we're going to start lab 3 right after lab quiz 1 is over. So we all got to finish at the same time. Any questions?
This is only for Monday lab, correct?
Yeah, only Monday lab today, and then only Wednesday lab on Wednesday.
Perfect.
Any other questions? All right, I went over a little bit, but that'll do it. See you on Monday lab and the lab quiz Zoom meeting.